Before antibiotics, a relatively minor infection could prove incurable or even deadly. Everything from paper cuts to childbirth had the potential to kill through bacterial infection. The accidental discovery of a moldy Petri dish in 1928 kick-started a 20-year-long journey to develop the world's first mass-produced drug that could clear a bacterial infection, penicillin. But why did it take so long, and how it was invented? In 1928, Alexander Fleming returned from a holiday to find mold growing on a Petri dish of Staphylococcus bacteria. He noticed the mold seemed to be preventing the bacteria around it from growing. He soon identified that the mold produced a self-defense chemical that could kill bacteria. He named the substance penicillin. Fleming published his findings and presented his discovery to the Medical Research Club. To his surprise, his peers showed little interest in his work. Undeterred, he enlisted the help of several chemists, all leading experts in their fields, to help purify penicillin from the mold. None were successful. Professor Harold Raystrick, a biochemist, an expert in fungal substances enlisted by Fleming, declared the production of penicillin for therapeutic purposes almost impossible. Penicillin was labeled a laboratory curiosity, and Fleming gave up attempts to purify it. Nearly 10 years later, in 1937, while investigating microorganisms and the substances they produced, Howard Florey and Ernst Chain uncovered Fleming's research and assembled a team of scientists to work solely on the penicillin project. Personality clashes between senior members of the team resulted in heated arguments over how to carry out the research. The ongoing disagreements within the lab, as well as the complexities and scientific challenges of the project, meant the team struggled immensely to purify penicillin from its original mold. After 3 years of trial and error, they developed a successful but painfully inefficient process that produced pure penicillin. The team finally had enough penicillin to start animal trials. In 1940, 8 mice were infected with deadly streptococci bacteria. Only the 4 also given penicillin survived. A paper they published detailing the experiment gained immediate interest, but the team were now severely limited by production. Gallons of mold broth was required to produce just a fingernail of penicillin. The team resorted to using bedpans, milk churns, food tins, and even bathtubs to store the broth. Eventually, special fermentation vessels were developed to hold the liquid. With penicillin now in successful albeit slow production, the Oxford laboratory had now become a penicillin factory. 6 women, known as the penicillin girls, were employed to tend to the fermenting broth and farm a few precious milligrams of penicillin from it every week. In 1941, the consequences of the team's production problems and shortage of penicillin became apparent with the first human trial of penicillin. Albert Alexander, a 43-year-old policeman, had developed a life-threatening infection from a cut. He initially showed signs of recovery, but the supply of penicillin quickly ran out and Albert's infection returned. He died 5 days later. The team had to turn to somewhat unpleasant measures to bolster their meager supplies. Around A of a dose of penicillin is excreted from our bodies in our urine and can extracted and recycled. Drive. Ethel Florey, a supervisor for the clinical trials, was regularly observed on the pee patrol cycling to patients to collect their urine. With their growing success, the Oxford team approached pharmaceutical companies to manufacture penicillin. However, with the Second World War in full swing, British industry was not capable of developing a new mass production process, so the team started to look elsewhere. In June 1941, Florey decided to take penicillin to the US in hope of finding a way to scale up production. In Peoria, Illinois, a new team was set up in the Department of Agriculture's Research Laboratory. They utilized their expertise in fermentation and designed new techniques using deep fermentation tanks to make the purification of penicillin as efficient as possible. The lab in Peoria had an abundance of corn steep liquor, a byproduct of corn starch. They discovered that when added to the mold broth, the yield of penicillin increased exponentially. The high concentration of sugars, amino acids, and nitrogen provided an excellent environment for mold fermentation. They started a global search for strains of mold with higher percentages of penicillin. Soil samples were sent in from around the world, but the solution was found closer to home. Mary Hunt, an assistant at the Peoria lab, found a rotting cantaloupe melon at a local market. The mold produced 6 times more penicillin than Fleming's original strain. Us pharmaceutical companies were initially reluctant to commit to large-scale penicillin production. However, by the end of 1940, the US joined the Second World War and demand for penicillin rocketed. By 1943, the US had sufficient penicillin stocks to satisfy the demands of the armed forces of the United States, as well as their allies. In 1946, penicillin became available for the first time in the US for public use. It transformed medicine worldwide and ushered in the age of antibiotics. But bacteria have evolved to become resistant to the original penicillin and many other antibiotics. Scientists are now in a race to find and develop new antibiotics.